Welcome to another episode from the Veterinary Research Channel. Today, we delve into the fascinating world of feline health, specifically focusing on a study investigating a condition known as congenital sensorineural deafness in purebred white Devon Rex cats. This research, conducted by Anne-Marie Cordes and colleagues from the University of Warmia in Missouri, in Poland, and Small Animal Clinic Kalbach in Germany, aims to provide valuable data on the occurrence of this type of deafness within this specific cat breed. Congenital sensorineural deafness, or CSD, is a hereditary condition affecting white cats, although the precise manner in which it is passed down through generations remains under investigation. It is understood that cats with blue irises are often considered to have a higher likelihood of developing this condition. This type of deafness is characterized by abnormalities and a loss of function in the delicate structures located within the inner ear, ultimately leading to hearing impairment. Much of our understanding of this disease in the past has stemmed from studies conducted on mixed breed cats within experimental colonies. However, the researchers in this study highlight that such data may not be directly applicable to understanding congenital sensorineural deafness in purebred cats. To date, only a limited number of studies have specifically examined this condition in purebred feline populations. Furthermore, these existing studies have often involved a relatively small number of individual cats, especially when compared to the extensive research conducted on hearing disorders in dogs. Hereditary sensorineural deafness can manifest in cats as a hearing deficit affecting one ear, known as unilateral deafness, or both ears, referred to as bilateral deafness. This hearing loss develops very early in a kitten's life, during the critical postnatal period when the structures of the cochlea are still maturing, meaning affected cats are often deaf from their first weeks. The underlying pathological process begins with the degeneration of the stria vascularis, a highly vascularized tissue in the inner ear. This degeneration then triggers a cascade of changes and a decline in the functional capacity of other essential components within the cochlea, including Reissner's membrane. Scientific literature suggests a strong link between the degeneration of the stria vascularis and a deficiency in functioning melanocytes. Melanocytes are specialized cells responsible for producing melanin, a pigment. In the inner ear, these cells play a crucial role in maintaining a high concentration of potassium ions in the endolymph, a fluid vital for generating the electrical potential necessary to convert external sound stimuli into electrical signals that the brain can interpret. The lack of melanocytes in white cats is associated with the presence of a dominant white gene, often denoted as W, which interferes with the normal maturation and distribution of melanoblasts, the precursor cells to melanocytes. However, the exact genetic mechanisms governing this inheritance pattern are still being elucidated. Previous research has put forward the hypothesis that congenital sensorineural deafness is linked to a mutation in the KIT gene located within the white spotting locus, which is also associated with the dominant white gene. This mutation is believed to involve the insertion of genetic material from the feline endogenous retrovirus, known as FERV1, within a specific region of this important gene. Recognizing and assessing hearing status in animals requires objective methods. Currently, the gold standard for evaluating hearing in animals is the analysis of results obtained from a procedure called the Brainstem Auditory Evoked Response Test, often abbreviated as BARE, which utilizes specialized electrodiagnostic equipment. This test measures the electrical activity in the brainstem in response to auditory stimuli. Given the limited data on congenital sensorineural deafness, specifically in purebred white Devon Rex cats, the primary objective of this study was to investigate the occurrence of this condition within a population of client-owned Devon Rex cats that were completely white in color. Furthermore, the researchers aimed to determine if there was any association between the presence of white pigmentation and congenital sensorineural deafness in this specific breed. To achieve these objectives, the researchers examined 40 client-owned purebred white Devon Rex cats. These cats were examined at 2 different veterinary facilities, with the median age of the cats being 19 weeks at the time of examination. Hearing status for each cat was carefully determined through the use of the Brainstem Auditory Evoked Response Test. Now, let us turn our attention to the findings of this important study. The results indicated that the occurrence of sensorineural deafness in the studied population of Devon Rex cats was estimated to be 10%. Interestingly, the occurrence of both unilateral deafness, affecting only one ear, and bilateral deafness, affecting both ears, was equally frequent, with 2 individual cats exhibiting each type, representing 5% of the total study population. The researchers also investigated whether there was any link between the occurrence of congenital sensorineural deafness and the sex of the Devon Rex cats in the study. Out of the 15 male cats examined, 2 were diagnosed with congenital sensorineural deafness, representing 13.3% of the male group. Among the 25 female cats, 2 were also affected by congenital sensorineural deafness, which constituted 8% of the female group. Statistical analysis revealed that there was no significant association found between the occurrence of congenital sensorineural deafness and the sex of the cats in this study. Notably, the 2 female cats diagnosed with congenital sensorineural deafness both exhibited unilateral deafness, while both male cats with the condition were bilaterally deaf. However, a direct comparison of the percentages of unilateral and bilateral deafness within the group of deaf cats did not show any statistically significant difference. Another key aspect of the study was to explore the potential association between the color of the cat's irises and the presence of congenital sensorineural deafness. Data on iris color was collected for all 40 cats in the study. 8 cats had both irises blue, and none of these cats were found to be deaf. 7 cats had 1 blue iris, and 2 of these cats were diagnosed with deafness. Among the 25 cats with both irises of a color other than blue, 2 were also found to be affected by deafness. When considering all cats with at least 1 blue iris, the occurrence of deafness was estimated at 13.3%. However, statistical analysis showed that in this study population of Devon Rex cats, there was no significant association between blue irises, whether one or both, and the occurrence of congenital sensorineural deafness. Within the group of 7 cats with only 1 blue iris, 1 cat was unilaterally deaf and 1 was bilaterally deaf. Interestingly, the researchers also documented a case where the owner of 1 cat had cleaned the cat's ears on the day of the brainstem auditory evoked response examination. In this specific instance, the test was conducted twice. Once on the day the ears were cleaned, and again 10 days later to investigate whether the presence of water in the external ear canal could influence the results. Upon comparing the recordings, it was observed that the presence of water in the ear canal appeared to decrease the amplitude of the recorded wave and lengthen the latency, or the time it takes for the electrical signal to be generated and recorded, in the left ear of the examined cat compared to the results obtained 10 days later. Now, let us delve into the discussion of these findings and their implications. The researchers emphasized that data on sensorineural deafness in purebred white cats remains scarce, with a majority of existing information pertaining to mixed breed cats, which may not be entirely relevant to purebred populations. While numerous previous studies have focused on the pathological changes occurring in the cochlea of the inner ear in white cats with congenital sensorineural deafness and the frequently cited association with blue irises, there is a relative lack of data specifically on the prevalence of this condition in different purebred cat breeds. The current understanding of sensorineural deafness prevalence in purebred white cats is largely based on 3 prior studies. However, the data from these studies has shown some variability, potentially due to differences in the specific breeds included and the total number of animals examined in each study. In this study of white purebred Devon Rex cats, the overall occurrence of congenital sensorineural deafness was found to be 10%. When comparing this to the overall prevalence reported in the previous 3 studies on white cats, which were 20.2%, 30.3%, and 16.7% respectively, the occurrence in Devon Rex cats appears to be lower. However, it is crucial to note that these previous percentages were derived from the total number of examined white animals across various breeds, not solely Devon Rex cats. In fact, the number of white Devon Rex cats included in those earlier studies was very small, with only 1, 12, and 6 individuals, respectively. The limited representation of Devon Rex cats in these earlier investigations makes direct comparisons of prevalence rates challenging. The researchers highlight that the small sample sizes in prior studies underscore the need for further research involving larger numbers of animals to gain a more accurate understanding of congenital sensorineural deafness prevalence in specific breeds. Currently, the available data on deafness prevalence in cats differs significantly from the more extensive data available for various dog breeds affected by congenital sensorineural deafness, where studies often include hundreds or even thousands of animals, lending greater statistical power to their findings. The researchers in this study acknowledge that their sample size of 40 cats is relatively modest, but express hope that collaboration among more facilities with electrodiagnostic equipment will enable the collection of sufficient data to match the level of understanding achieved in canine populations with this condition. A notable finding of this study was the lack of a statistically significant association between deafness and blue irises in the examined Devon Rex cats. This finding aligns with one previous study that also found no significant association, but contradicts the results of two other earlier investigations that did report a significant link. The researchers suggest that these discrepancies in findings could be due to the different number of individuals examined across studies and the variety of breeds included in those broader analyses. Furthermore, they propose that variations in the genetic makeup and reproductive strategies within the studied cat populations could also contribute to these differing results. In their study, none of the 8 cats with 2 blue irises were deaf, while 2 cats with 1 blue iris and 2 cats with non-blue irises were diagnosed with congenital sensorineural deafness. While the occurrence of deafness in cats with at least 1 blue iris was estimated at 13.3%, the study found no difference in the association of deafness between cats with 1 or 2 blue irises, a finding consistent with previous research. When comparing their results to a previous study that examined a similar number of cats, the researchers noted a significant difference in the prevalence of congenital sensorineural deafness. That earlier study found a prevalence of 43.9% in Norwegian forest cats, which is considerably higher than the 10% observed in the Devon Rex cats in the current study. The researchers propose that this substantial difference between the two breeds could be attributed to various factors, such as differing reproductive practices or the inherent randomness in clinical studies. But it may also indicate a genuine difference in susceptibility to congenital sensorineural deafness between Devon Rex and Norwegian forest cats. Just as significant variations in deafness prevalence exist among white pigmented dog breeds, similar breed-specific differences may exist in cats. A previous study using statistical modeling indicated that Norwegian forest cats were significantly more likely to have congenital sensorineural deafness compared to other cat breeds in their study population. Additionally, that study suggested that cats with at least 1 blue iris were more than 3 times as likely to be deaf compared to Norwegian forest cats with non-blue irises. In contrast, the current study on Devon Rex cats found no such association between deafness and the presence of at least 1 blue iris. The researchers acknowledged that the limited sample size in their study could be a contributing factor to the lack of an identified association. Regarding the potential influence of sex, this study found no association between the occurrence of congenital sensorineural deafness in Devon Rex cats and their sex, which is consistent with findings reported in previous studies involving a larger number of cat breeds. In this research, both female Devon Rex cats with congenital sensorineural deafness were unilaterally deaf, an observation also noted in a prior study. In the study group of 40 Devon Rex cats, 2 exhibited unilateral deafness and 2 had bilateral congenital sensorineural deafness. The percentage of unilateral and bilateral deafness in the examined population was therefore the same, estimated at 5% in both groups. Among the deaf cats, there was no significant difference in the percentage of unilateral versus bilateral deafness. The researchers point out that data on the differences in the occurrence of unilateral and bilateral deafness in cats is limited and inconsistent across the existing literature. While 2 previous studies reported similar percentages of both types of deafness, another study indicated that unilateral deafness was twice as common as bilateral deafness. Due to this lack of definitive data, the researchers emphasized the need for further investigation into the occurrence of these different types of deafness within the broader population of cats affected by congenital sensorineural deafness. Furthermore, they highlight that the incomplete understanding of the precise mechanisms of congenital sensorineural deafness inheritance, particularly the genetic factors determining unilateral versus bilateral involvement, underscores the necessity for research involving a larger number of affected cats. In conclusion, the findings of this study suggest that the occurrence of congenital sensorineural deafness in white Devon Rex cats may be lower than previously reported in studies examining the prevalence of this condition in purebred white cats in general. Another notable difference compared to some of the existing literature is the lack of a statistically significant association between the occurrence of deafness and the presence of blue irises in this specific breed. The researchers propose that these discrepancies may arise from numerous differences between the compared cat populations, as well as the relatively limited number of animals examined in all of these studies. However, they specifically highlight the contrast in congenital sensorineural deafness occurrence and its association with blue irises between Devon Rex and Norwegian forest cats as particularly noteworthy. These observed dissimilarities strongly support the need for further research focused on the occurrence of congenital sensorineural deafness in individual cat breeds, where the dominant white gene is present. The researchers also acknowledge an increasing awareness among breeders in Poland regarding congenital sensorineural deafness and a growing interest in reducing or eliminating the disease within purebred cat populations, as evidenced by a recent resolution from the Polish Phenological Federation. They believe that this increased focus on the issue, including the mandated hearing testing using brainstem auditory evoked response for all white cats intended for breeding, may contribute to the discovery of currently unknown inheritance mechanisms and more accurate prevalence rates in the future. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.